Okay, good morning, everyone. And first of all, I would like to thank the organizers for this possibility of presenting the data of my research. That, as you can read from the title, is about the biology of the oligodendrocyte precursor cells and their differentiation. The oligodendrocyte precursor cells are the cells responsible for the developmental myelination and the myelin repair. They have been recently discovered, and we know that they account for the 5-8% of the total cells in the mature central nervous system, recognized by the marker Ng2. They perform the myelination and the repair of the myelin, so the remyelination. They are proliferating cells, as you can see from the double staining of the proliferation marker MCM2 with the Ng2. And new OPCs, as we will see after in the presentation, can be generated from the neural stem cells. During the development, OPCs derive from the radial glia. They proliferate and migrate to the new axons. They differentiate in mature oligodendrocytes, wraps to the unmyelinated axons, and generate the myelin sheaths. This process is controlled mainly by the thyroid hormone and occurs mainly at the end of the gestation and postnatal. Some of the OPCs generated during the developmental myelination remain in undifferentiated state in the CNS, in a quiescent state, generating the pool of resident OPCs that we found in the adult brain and spinal cord. They are responsible for the physiological turnover and the repairment of the myelin. And they can be, in fact, activated by different stimuli deriving from the myelin damage, such as inflammatory cytokines, myelin debris, or conditions, pathological conditions like hypoxia ischemia. And once activated, they start again to proliferate and migrate to the demyelinated axons, where they differentiate again in mature oligodendrocytes. And to be noted is that this is the only nervous system capacity that leads to a totally functional and anatomical restoring. Thyroid hormone is the main regulator of both processes, and its action is locally regulated by specific transporters and enzymes that can activate or inactivate the hormone. These are the deiodinases enzymes, which remove the iodine atoms from specific positions. We have the deiodinases 2, which activate the T4 in T3, that is the active form of the hormone. And then we have the deiodinases 3, which inactivate both the T3 in T2 and T4 in reverse T3. The equilibrium of the hormone in the blood circulation is unbalanced to the inactive form, which account for the 80% of the whole circulating hormone. And already this equilibrium in these levels can result in pathological conditions such as cretinism during development, or also can occur during adult life with hypothyroidism. TH are then transported in the blood circulation inside the cells by specific proteins, and we know that it is the intracellular level of the hormone that is fundamental for the OPC differentiation. In fact, when, for example, the transporter, the MCT8, is not working properly, like in the AHD syndrome, there will be a problem in the myelination and not a proper myelination, leading to psychomotor retardation and hypomyelination disorder. The genomic active form, the T3, works as a ligand for nuclear receptors. So it activates or inactivates specific gene targets. Nuclear receptors are a superfamily of transcription factors. They are composed by a DNA binding region which bind the ligand and also bind the co-regulators, and also a dimerization region. because they usually work as homodimers or heterodimers with other nuclear receptors. The thyroid receptors mainly perform the heterodimerization with the RXRs, and they are usually bound already to the DNA target. And when the thyroid hormone bind to the receptor, they change the conformation and recruit co-regulators to activate or inactivate the transcription. As proteins, transcription factors, and for their ligand-dependent activities, nuclear receptors show a high druggable nature. They are seen as therapeutic targets for different diseases, and they are already a target of different therapies. But this is the genomic action of the thyroid hormone, which is the main regulator of the OPC differentiation. However, T3 exerts also a non-genomic action mediated by different receptors and with a central role of sonic hedgehog in this process. And moreover, other characters are orchestrating the complex differentiation machinery as growth factors, neurotransmitters, and epigenetic modificators. Different pathological conditions may interfere with both developmental myelination and remyelination in the adult, as in the examples shown in the slide, like neonatal hypoxia ischemia or different pathologies in the adult life. In this sense, the emblematic pathology is the multiple sclerosis. In this pathology, the myelin is disrupted, and the OPCs are recruited in the initial phase of pathology. They can restore the myelin sheath. However, with the progression of the pathology, this process progressively fails. And OPCs reaches the demyelinated axons, they proliferate, but they are not able to differentiate in mature oligodendrocytes. This pathological process is named the OPC maturation block and is seen as one of the main causes of the remyelination failure in MS. We are able to replicate the pathology in vivo using the experimental allergic encephalomyelitis model. And here you can see a section of spinal cord marked with a fluoromyelin to visualize the myelin, and the control conditions, and in the acute phase and in the relapsing remitting phase of the pathology, where the myelin is progressively disrupted. The objective of our studies of the last years on this topic is the investigation of the pathological components of the demyelinating diseases, such as inflammation and hypoxia ischemia conditions, to find the mechanisms underlying the OPC differentiation block. And the main cell target of the therapeutic strategies is, of course, the neuron, but this pathological condition affects also the other cells, inhabitant of our CNS, which participate actively to its function. And it is fundamental to understand the vulnerability of the cells and include them in the therapeutic strategy, because it will be useless to protect the neuron without protecting also the other cells participating in the CNS physiology. So we are selecting the most suitable in vitro model, and we are searching for the molecular link between inflammation and the OPC differentiation block. We are studying the physiological differentiation of both fetal and adult OPCs, studying also the role of nuclear receptors in these processes. To dissect the components of the pathologies that may affect the OPCs, we use an in vitro model generating mature oligodendrocytes from fetal and adult neural stem cells. The culture contemplates the production of neurospheres from fetal forebrain and adult subventricular zone, keeping them in suspension and in proliferation conditions by using specific growth factors. That after the first passage, it's included the PDGF, which start starting a differentiation through the oligodendroglial lineage, producing the so-called oligospheres. When they are big enough, they are dissociated and seeded on a laminin based coating, and after 3 days, the OPC's medium is removed and substituted with the one containing thyrodormon. And this allows the production of mature oligodendrocytes expressing specific markers like MVP or SYNPase, passing through the all differentiation steps. And the cells produced by both fetal and adult tissues resemble in vivo conditions. In fact, as you can see from the pictures, if the thyrodormon is not present, they are unable to differentiate. For this reason, we investigated the locally regulated T3 metabolism in the EA model, finding that both in acute and relapsing remitting phase, there is an increase in the expression of the inactivating enzyme of thyrodormon, the deiodinases 3. And that is also expressed in the infiltrating cells of the immune system, as proven by microdissection experiments. This observation drives us to elaborate the hypothesis that the inflammation microenvironment leads to an increase in the expression and activity of the deiodinases 3, which in turn leads to a reduction in the availability of the thyrodormon locally, and so to the OPC differentiation block. To prove it, we exposed in vitro the OPCs to a mix of cytokines identified in the in vivo model, and the exposure produced an increase in the cells expressing D3, which lead to a strong reduction of the percentage of mature oligodendrocyte and an increase of the precursors, which is a picture of the expected differentiation block. However, to direct link the cytokines exposure with the block in the differentiation mediated by deiodinases 3, we treated the cells with eupanoic acid, which is a deiodinases inhibitor, and the treatment resulted in a restoring of the differentiation, confirming our hypothesis. We then moved our attention to the physiological differentiation of these cells. Seeing the importance of the nuclear receptors on the differentiation process, we analyzed the expression of the wall nuclear receptors and co-regulators using a gene expression PCR array. And we found that the adult, compared to the fetal neurastem cells, they have upregulated profile, gene expression profile. And in particular, we focus on two nuclear receptors that are very important for the OPCs, because we found that in the adult we have a decrease in the expression of the RXR gamma and an increase of expression of thyroid hormone receptor beta. So what is the consequence of this different expression in terms of OPC differentiation? So when fetal and adult cells are physiologically differentiated by thyroid hormone, we found that the adult are less prone to differentiate, reaching a reduced yield of mature oligodendrocytes and an increased percentage of precursors at the end of the differentiation period. And moreover, at this stage, also the RXR gamma gene is still less expressed in the adult, pushing to raise the question, so what is the role of the RXR gamma in physiological differentiation and pathological conditions? To answer this question, we used a knockout mouse lacking the RXR gamma gene, and we replicated the OPC's culture. And as you can see from the pictures, we found a strong reduction in oligodendrocyte differentiation, both in fetal and adult cultures. Of course, more in the adult that were already lacking some differentiation potential. And this differentiation block is very similar to the one produced by inflammation. So when RXR gamma is missing, OPC's differentiation maturation is impaired. And as you remember from the introduction, the thyroid hormone is necessary, is the main signal to start the differentiation, because it push out the OPCs from the cell cycle, starting the expression of the differentiation genes. So we analyzed the cell proliferation, quantifying the OPCs identified by PDGF-alpha receptors in replication, so double positive for Ki67. In wild-type cells, as you can see, when we give thyroid hormone for 24 hours, we have a reductions of the replicating OPCs. In the knockout, in basal conditions, we have already more OPCs replicating. And when we give the thyroid hormone, it totally fails to stop cell proliferation. So we hypothesize that when RXR gamma is missing, OPCs are unable to respond to thyroid hormone-induced cell cycle exit. It also fails to regulate specific direct genomic target, such as P27, c-MYC. As you can see, they are regulated only in wild type, but not in the knockout. And the PDGF-alpha receptor, which is down-regulated in the wild type, is going in a totally other direction in the knockout. And this block in the differentiation is also evident when at the end of the differentiation phase, the knockout cultures are full of precursors. Again, as in the inflammation-induced OPC differentiation block. To go deeper in the mechanism of the differentiation, to try to elucidate the role of this nuclear receptor in the process, we performed a series of RNA-seq experiments. First, as it's possible to see by the graph showing the number of genes upregulated and downregulated, we have almost 250 genes differentially regulated between knockout and wild type, so they have a really dysregulated profiles. And if we take a look in the same genotype, after the thyroid hormone administration, we can see that RXR gamma cultures have half of the genes regulated by thyroid hormone. And again, we expected this because these cells were not able to respond to the genomic action of the thyroid hormone. Increasing the magnification in the downregulated genes, we can see that most of them are related to myelination. Here we have the main cluster, and all these genes are related to myelination. This is a protein net analysis made by String Software, based on the more than twofold regulated genes, and here there is the central core made by these myelination-specific genes, but also all the other genes in the net are somehow related to the differentiation of OPCs. But more interestingly, when we compare the expected impairment in the response of the T3 with the upregulated genes, one of the main clusters is composed by the sonic hedgehog and related genes. This is part of the non-genomic action, so it seems that when RXR gamma is missing, we have an unbalance between the non-genomic action, which is upregulated, and the genomic action, which is downregulated. So seeing the effect of the RXR gamma knockout similar to the differentiation block demonstrated in inflammation model, resembling what takes place in some demyelinating conditions, we then try to interfere with this suggested imbalance to restore the differentiation in RXR gamma KOPCs, also because we know from also other groups that RXR gamma is fundamental for the remyelination. Since the sonic hedgehog pathway was upregulated, and since we have different molecular keys to interfere with and block the sonic hedgehog pathway, we use two different strategies. So cyclopamine, which acts in the first phases of the pathway, blocking the smooth net protein, and GAN61, which acts at the end of the pathway blocking the GLI signaling. First, we showed that treating the KIO cells with both inhibitors, so GAN61 and cyclopamine, this restored their capacity to respond to thyroid hormone and exit the cell cycle, as you can see from the decrease of the proliferating OPCs only in the treated cells when, of course, thyroid hormone is present. After we directly looked into the differentiation and maturation of the cells, and interestingly, the treatment with both inhibitors was sufficient to totally restore the differentiation, as you can see from the increase of the mature oligodendrocytes and decrease of the precursors in the knockout. And however, in the wild type, the effect was totally opposite. So we have also a decrease in the differentiation and even a decrease in the precursor. So something was wrong in the wild type when we interfere with the physiological sonic hedgehog, and this suggests that a proper level of sonic hedgehog is necessary for a proper differentiation. So it's not a black or white stories, but there is a lot of shades of gray between them, this equilibrium. We analyzed also the 3D structures of the mature oligodendrocytes by confocal images and IMARIS software, proving that also the maturation of the oligodendrocytes follow the same response to sonic hedgehog inhibition. So we see restoring of the morphology of the mature oligodendrocytes in knockout, while in the wild type the maturation is impaired. And we are now studying the direct target of the RXR gamma by CHIP PCR and ChIP-seq to identify where the RXR gamma is bound in the DNA of the wild type, and so to try to identify what is the responsible of this dysregulation in knockout. And we are also trying to identify the other pathways involved, because, as I said before, this is a very complex machinery. So the T3 is the main character, but we have also other signals. And to conclude, we show here that chronic inflammation blocks the OPC differentiation by reducing the availability of thyroid hormone. And we asked if RXR gamma nuclear receptors is involved in these inflammation-mediated effects. And in fact, the absence of RXR gamma resembles the inflammation-mediated OPC differentiation block, leading maybe to an impairment of thyroid hormone signaling. Since there is an imbalance between T3-mediated genomic and non-genomic action that may answer to our questions. So we are now identifying the targets of the dimers of the RXR gamma with the thyroid hormone receptors, and we are trying to find a link between RXR gamma target genes and sonic hedgehog pathway to find if the RXR gamma can be a putative therapeutic target for demyelinating diseases. And at the end, I would like to thank the groups working on this project in Bologna, in Italy, under the direction of Professor Laura Calzà and Luciano Giardino, and in the University of Strasbourg under the direction of Wojciech Krezel. And thank you for your attention. If you have any questions, this is also my email. Thank you.